often we still have to get the protein to the right place after it's been made. So those are all important. So really, strictly speaking, this is just about the synthesis of the correct polypeptide chain with the correct polypeptide sequence, and then we'll look forward after that to the initial regulation a little bit about how those liposomes are getting the protein as well, doing the correct post-modification and the correct location. I want to emphasize something really, really important. A lot of emphasis and focus, even in this PowerPoint and even in this lecture, goes to the idea of making a peptide bond. And as we saw last semester, and as we'll see again, there are some chemical challenges associated with that. But that's not the only thing that the ribosome has to do. In fact, if you think about it, if the only thing to do was to make a peptide bond, that was the only challenge that we had, the chemistry should be rather simple. The ribosome is this big complex made up of lots of protein, lots of RNA. 35, I think, percent of the dry mass of your cell is just ribosomal component. And yet the chemistry itself is really very, very easy making the peptide bond. We just kind of have our typical challenge to overcome, right? Not having a good leaving group. And so even though that is one of the things that the ribosome does, if that was all the ribosome had to do, things wouldn't have to be so complicated. We would not need to have this enormous machine made up of all of these polypeptides, all of these RNAs, all these various components coming and going, if our major challenge was to make a peptide bond. Making a peptide bond should be very, very straightforward. We saw, in fact, very relatively simple chemistry on this respect last semester when we did peptide synthesis. The ribosome has to do the same thing. I'm not saying it was the easiest chemistry, but really it was a matter of adding the good leaving, right? And so it's really critical right at the beginning that we emphasize the goal that if you're ever going to build something, or at least build something useful, you really have two important jobs, not one. One of those jobs is construction, making that thing that you want to make, which in this case, of course, is peptide bonds and making a protein, right? But that's not the only important feature of construction. The other important feature of construction is being able to read your plans and effectively turn those plans into the thing you want to make. You don't try to build something until you know not only how to have the instructions for that thing, but also how to interpret the instructions for those things. And as we'll see, the major feature of the ribosome, in fact, the major source of energy investment in the ribosome, doesn't turn out to be making peptide bonds. It's making peptide bonds with the correct peptide sequence. Now, last semester, when we talked about peptide synthesis, that wasn't a problem. It wasn't a problem because we were in charge, right? We were deciding which amino acid to add next. Put in an alanine, do your washing and shaking steps, your key protections, put in a valine, so on and so forth. But of course, there's no master regulator watching over the synthesis of polypeptides inside of the cell. So if there's not going to be someone like playing the role that we did in peptide synthesis, there's going to have to be a machinery for making sure that the instructions are read correctly. Not just that peptide bonds are made, but that peptide bonds are made in the right order to give us the primary sequence we want. And so what's really amazing about the ribosome is not that it makes peptide bonds, but rather the idea that more energy actually goes into getting the right peptide bond made than actually goes to making a bond. We actually devote more energy to the instruction step than we do to the assembly step. And that's really what the ribosome is there for. So again, the chemistry, at least this is in the past how we've drawn it out, just having two amino acids coming together. This is the way it's typically shown in the first or early chapters of a biochemistry textbook or even a biology textbook. But you remember, it's a simple nucleophilic attack, which this perspective we early on recognized couldn't possibly be valid. There's just not a good leaving group here for that. And we saw that not only in the context last semester of the organic chemistry, but in terms of thermodynamics as well. We just went and calculated the standard free energy of actually making a peptide bond by taking the differences in the free energy of formation of the reactants from the products. Here is all the math. Here are the numbers that we actually looked up. And when we did this calculation back in Biochemistry 1, you'll recall that we saw that the delta G naught under standard conditions anyway was about 15 kilojoules per mole, which translates to an equilibrium constant of less than 0.0003, which means that making a peptide bond itself is not favorable. So we go know thermodynamically it's not favorable. We can understand from the perspective of organic chemistry why it's not favorable. But again, we know now how to make things favorable that are usually not. And why are things not normally favorable? At least when we're doing these biosynthetic reactions, we know we just generally don't have a very good leaving group. There are other issues as well, of course. The entropy of two glycines is much higher than the entropy of a glycine together. But our real challenge is making a good leaving group. And so that's one of the specific challenges that the ribozyme has to overcome. Of course, we already know how to overcome that challenge. We're just going to use coupling. And we could always just use coupling right to ATP. We've done it before in a multitude of different ways when we made phosphodiester bonds. We could, in theory, do the same chemistry with peptides and be all done. And in fact, that is what we're going to do. We are going to, in fact, couple the synthesis of peptide bonds to the hydrolysis of ATP via making a good leaving group. The only real problem we have with this is that the enzyme that we're going to put in charge of doing all this doesn't use ATP as a substrate. So we're going to have to kind of see how nature manages to use coupling to ATP by an enzyme that does not accept ATP as one of its substrates. So that is kind of the energetic challenge we have, but I think we already kind of know how to overcome that, at least a little bit organic chemistry-wise. The real big problem, as I've already stated, therefore, is not making peptide bonds in the right order, establishing the correct primary sequence, ensuring that we have a sequence that is repeatable and reproducible as long as that mRNA message is the same. And again, most of the energy that we are going to input into this process is actually going to be devoted to getting the order right. The actual making of the peptide bonds will take some energy, of course, but not as much energy as the amount of energy to make sure that the sequence is actually correct. So what are the challenges, if we can kind of summarize this all up now that we have? We know that we have to catalyze a thermodynamically unfavorable reaction, but whatever. Coupling here, coupling there, we keep coupling ATP. Find a new way to do it. That's something that we can imagine is possible. We only have one copy of the template, right? So this was an issue that we faced a bit when we made RNA, but certainly we didn't have when we looked at making DNA, right? When we replicated DNA, there was always another molecule around to make sure we got it right. There was not a molecule around, of course, to check the mRNA against, and certainly there's not going to be anything else to check the protein sequence that we're making against, because we only have one mRNA to work with. So that is a bit of a problem. The ribosome does not have an error-correcting site, so there must be some way to ensure this, getting the right sequence without error correction. That's something we need to look at. And really, right from the beginning, since we're talking about information, we have a 4-letter alphabet to work with, right? Just 4 letters. And we need to translate that into a 20-letter alphabet. And so here comes, right, the real significant feature of the ribosome. How do we translate a 4-letter alphabet into a 20-letter alphabet and do so with high information fidelity, especially when we really don't have any error checking possibilities? Okay, so again, here we go. Our question stated like this, since we mostly like talking things in terms of energy. How do we use coupling for an enzyme that doesn't take ATP as a substrate? How is it going to couple to ATP hydrolysis? And also, how are we going to use energy to make sure that we get the sequence right for an enzyme that lacks fundamentally any error correcting capacity? Yes, here we go. So, before we go forward, there is a cofactor that I need to introduce to you. We looked at it really briefly before, so I want to show you this again. We're actually only going to see this cofactor really once in this particular lecture, but we're going to see it get used a couple times in some other metabolism lectures. So I think now is a good time. This is the cofactor tetrahydrofolate, or THF, which just wipe out tetrahydrofuran in your minds. I mean, some of the organic chemistry focused people here will write tetrahydrofuran. No, tetrahydrofolate. I'm sorry that the abbreviations is very similar to something that you saw in organic chemistry. Anyway, what does this cofactor do? This is the cofactor of one carbon atom in a variety of oxidation states. All of this that you see here, all of this big mash of atoms here, has been assembled to carry out one carbon. And you might wonder, well, that's It's a lot of carbon to devote to one carbon, right? I mean, all those molecules that carry that one carbon. But if we can go all the way back to biochemistry 1, right back to the beginning, we stated that one of the critical features of life on Earth, in fact, one of the necessities to having biochemistry, is just how rare carbon is in our environment. Not a lot of carbon in the crust, right? Not a lot of carbon for organic matter for living organisms to deal with. Which means that if you've captured a carbon atom, you want to hold on to it, you want to use it for as long as you possibly can before it has to go out into the waste bin to be recycled again. And so this is the cofactor that really ensures that sort of ability to retain single carbon atoms. These carbon atoms, and we'll see how later, will have been broken off of other molecules, quite often amino acids, and in the process of fragmenting them into different pieces, single carbon atoms will be generated. And those carbon atoms can be located or attached to either the N5 or the N10 nitrogen, or in some cases both. So that is one of the important features that this cofactor has. It can hold on to a single carbon atom that has been liberated in a variety of the metabolic processes that we will look at later. But that's like not the only thing that THF is important for. Because sometimes, for example, you might need to have carbon atoms for biosynthetic purposes in different oxidation states. You may need highly oxidized carbon. You may need more of a reduced carbon, right? Methyl group versus having a carbonyl group. And the other thing that this cofactor can do is allow those oxidation and reduction reactions to happen on the carbon while it is attached to them. So if you, for example, were to scavenge an oxidized carbon, like you see here with N10 formal THF, but you need a methyl group, it can be reduced while it is still attached to the THF cofactor. So it allows you to hold on to carbon atoms in different oxidation states without those carbon atoms which are generated in metabolic processes from just being discarded. So it's a very efficient, you could think about an environmentally ideal cofactor to have. We'll only see it once appearing in this lecture. We'll see it more later on. But this seemed like a good time to introduce this cofactor now. THF, which can hold on to carbon in the N5 or N10 positions in a variety of different oxidation states, and then donate them to other biosynthetic pathways as they are needed. Okay, so what do we need to know about protein biosynthesis before we get started? It's going to take at least 70 different proteins, probably more than that, but at least 70 well-defined proteins. 20 are going to be required alone just to get the amino acids ready to go. Okay, so 70 ribosomal proteins, 20 before it even hits the ribosome. So we're up to a pretty high protein content. Now, 100 after this lecture, at least, are going to be required to do the sorts of post-translational modifications, but those are outside of this PowerPoint. Those are next. So our protein count has gone really high and we barely even got started. More than 40 different types of ribosomal RNA, most of which are actually going to be in the shadows. We're not going to see them, but you should appreciate they were there because none of this chemistry is going to happen without them. So at least 40 different types of RNA molecules. And these are big RNA molecules, right? So these are ones that are making up the bulk of the mass of the RNA that you actually have. Extraordinarily energy and intensive. 35% of your dry mass in carbon, nitrogen, and oxygen and hydrogen. Your solid mass, once you've taken the water out of a cell, is just going to come from the protein biosynthesis, mostly mass of the ribosomes themselves. Very enormous. It's going to happen really fast. Roughly 100 amino acids an E. coli ribosome can do in approximately 5 seconds. But that is not uniform. Some proteins take longer depending on their sequence. A few can take actually shorter. And it does not necessarily happen continuously. So we can now actually see individual proteins coming off of ribosomes, or at least study and measure that. And they seem to happen in bursts. But there is some sequence difference. But on average, if you want to know how long this takes to make about 100 amino acids, roughly 5 seconds is a pretty good estimate. Okay, so we talked about the transcriptome being the total combination of all the possible RNAs that are present inside of a particular biological entity. We also have a proteome. A proteome effectively that's the same thing with the polypeptides that might be present. All of the proteins that are present in some particular biological entity. Of course, you could also have a particular type of cellular proteome, but often we're just thinking about an organism's proteome. So viruses, for example, depending on their size and molecular complexity, this is anywhere between 3 and 1,000 different proteins they have in their proteome. Looking at a prokaryote, the simplest ones, about 500, all the way going up to 10,000. Ours, believe it or not, we do not know. We know a roughly estimate on how many protein-coding genes that there actually are. That's somewhere typically given in the 20,000. But between all of the different types of post-translational modifications, the possibilities of multiple genes being coded for by the same RNA molecule, depending on things like poly A choice and alternative splicing, we really don't know. I mean, it's a lot. I think it's pretty much all we can say about our human proteome. It's certainly much bigger than that of the prokaryotic proteome. And then we have something called the dark proteome. The dark proteome represents all of those proteins in an organism, many of which are actually eukaryotic, that we actually have no idea, or at least no proof for what they actually do. In some cases might have some idea what they might be doing based on some sequence or potentially structural conservation, but we're not actually sure. And remarkably, there are some estimates that up to 50% of the proteins that are present inside of your body, we still cannot conclusively identify what they're actually doing. So the dark proteome, this kind of hidden, mysterious proteins that are there, we know they're there, but it's not clear what they do is actually can be quite substantial. And again, just showing you how the idea of how you can start with only 20 or maybe 25,000 genes, but through alternative splicing and poly A choice, again, coming up with more than 100,000 different types of mRNA transcripts. And then when you add things like post-translational modifications, one of the ideas we talked about last semester was that a single polypeptide chain can actually be hacked up into multiple pieces, each of which are functionally independent of one another. There could be far more than 1 million actual proteins. So very efficient way of coding, right? Just 20 to 25,000 genes in excess of probably 1 million different functional entities which we call proteins. Okay, so let's step all the way back to the beginning, because I just love this experiment, and I know we're running late, but I cannot not tell you about the cool experiment. Like, how do we know about this machine we keep calling the ribosome? Like, how was this actually discovered? By like, one of just the coolest possible experiments. I mean, this had to have been proposed in the day before like anybody was really checking. Because like, here's what some scientists did. They went and got some rats, and they injected radioactive amino acids into them. And I mean, that's like the start of every good science fiction story, right? Like, some radioactive amino acids got injected into the rat. Anyway, the rats didn't do all that well, obviously, not just because they had radioactive amino acids injected into them, but because they were being overseen by scientists who wanted to know what was going to happen to those radioactive amino acids. But what they found was really interesting, and it depended on when they looked. If you let the rats live for a couple days, the amino acids just kind of ended up all over the place. Not surprisingly, the rat has a lot of protein, those proteins kind of got distributed. But intriguingly enough, if they killed the rats a short time, a very, very short time after the amino acids were initially injected into them, they seem to be found in these really dense granules, really concentrated in these granules that when placed in a centrifuge tube would sink right to the bottom. And very little of the amino acids would be found any other places. These granules are what we now know as ribosomes. And you can see some of them here in the electron microscopy images. Very, very large, these large kind of dark spots that are kind of literally just images of the cell. These are actually ribosomes that are associated with the endoplasmic reticulum. We'll see later that those are ones that are specialized for making membrane proteins, but they're kind of functionally the same. And these are very large, very dense, so they always will spin to the bottom of the centrifuge tube, and that's where the radioactivity was concentrated. The other discovery, which at the time really wasn't very well understood, was that if you took these radioactive amino acids, and you actually purified and took some of them out, went through all the purification steps, instead of finding free amino acids, you found amino acids that seemed to be chemically linked to another molecule, which turned out to be a nucleic acid. So this is a separate set of experiments that are being done now, but these amino acids didn't seem to be just kind of randomly floating around diffusely, but rather they were covalently connected to amino acids. And now, of course, we know that these RNAs, these RNAs are now what we call tRNAs, right? So these two pieces of evidence really provided us much of our understanding, or at least the foundation for the understanding of how this whole process works. the finding that amino acids would quickly concentrate in these very dense granules that today we call ribosomes. And the fact that those amino acids, rather than just be floating around, are actually covalently linked to nucleic acids, which today we know as tRNAs. Again, we've seen this slide before, so this is just by mass. These tRNAs, I just wanted to show you that there is an enormous number of them, even if they are not necessarily very heavy. So if you're wondering how could we have found so many tRNAs connected to so much amino acid, well, cells are just kind of loaded up with these things. So it wasn't too surprising that tRNAs weren't that difficult to discover as soon as someone went looking for the amino acids that they were attached to. Anyway, the original thought for how this must work was pretty much similar to the rest of the biochemistry we've covered before, right? There must be some machine that grabs an amino acid and takes another amino acid and connects those amino acids, and that's how polypeptides get made. But Francis Crick kind of figured out that this was sort of a very inefficient way, or at least considered the possibility that this was sort of an inefficient way to do so, right? Because you would need to kind of have one enzyme that could somehow sense what the amino acid is and also be sensing what the RNA was reading, the codons that it would be reading out, and somehow know to connect these all together. And that's kind of complicated for just one sort of biological enzyme to do. Certainly not impossible, but the idea that either you're going to have a lot of different machines for each different combinations of amino acids, or perhaps one machine that could recognize all these different amino acids and the RNAs and bring this all together, just didn't seem plausible to Crick. So he instead proposed this idea called the adapter hypothesis, which would be that the amino acid that was going to be connected or read, as we would now say, would be attached at one end, and the other end would be able to recognize the genetic coding sequence for it. And if you just kind of line these things up on the RNA, that would sort of be how the right amino acids actually got together. So instead of the amino acids somehow directly associating with the message that was being read, there would be some sort of adapter molecule. And so the connecting machinery could be up at one end and be fairly general, not necessarily worried about which specific amino acid it was, and the reading machine could be down here to read out the correct part of the sequence. And so this is what Crick proposes as his adapter hypothesis. Today, of course, we know, yes, the adapter is in fact the tRNA molecule that had been independently discovered by those previous experiments. But it was really, again, this insight into how impractical it might be to somehow amino acids or even an amino acid binding enzyme just directly recognizing the mRNA, and how it would be better to separate these two things out, the recognition process on one side and the connecting to everything together process on the other side, which could then be generalized. So the chemistry that was taking place under Crick's model would not be dependent upon what amino acid it was, right? It could connect then any two amino acids that just happen to be next to each other on the message. And so that was Crick's suggestion, and of course we now know it's correct, and we now know that these adapters are what the tRNA was discovered earlier. The next question, as people were trying to figure out how to read the message, is what type of a code is this? So they quickly settled on 3 bases instead of just 2 bases or 1 base. We know it can't be 1 base, right, because there's only 4 letters, 4 letters in the genetic code, and there are, of course, 20 in the amino acid code. So it can't be 1-to-1. Even 2-to-1 can't do it, right? If you just use a 2 combination, you're not going to have enough. 3 is actually a bit more than you need, and 4 is excessive, right? So if you think about all the permutations of a 4-letter alphabet, 1 doesn't give you enough amino acids, 2 doesn't give you enough possible codes for amino acids, 3 gives you a little more than you need, and 4 is way excessive. So really, with no experimental evidence to go on, they just settled on the idea, let's start working on this under the assumption that it's 3, and let's hope we're right, because we're about to do a lot of biochemistry here. And of course, we now know that it is 3. We do read a language of 3 bases to make a code for a single amino acid, so that was a good idea. The question is, how does this code get read? And if you have a code, it could be 3, but really of anything more than 1, there are 2 possibilities. One of these is an overlapping code, and one of these is a non-overlapping code. So the non-overlapping code we can see on the top, that code says that each 3 bases corresponds to what we call a codon and would code for a particular amino acid. So 1 amino acid, 3 bases, the next 3 amino acids are the second base, the third 3 amino acids, the third 33 nucleotides on the third amino acid, and so on and so forth. This is what we call a non-overlapping code. The advantage of a non-overlapping code is that you can change any one of these bases, right, within a particular codon, and you're only going to change a single amino acid. So it's fairly resistant, right, to the types of errors that might take place during all of this genetic processes of replication and then transcription and so forth. The second possibility was an overlapping code. Now, an overlapping code, as you could see, still uses 3 bases for a codon to code for a single amino acid, but it's always shifting over by 1, right? So the first 3 bases here would code for 1 amino acid, and then shift over by 1, so base 2, 3, and 4 would code for the second amino acid, and then 3, 4, and 5 would code for the third amino acid, and so on and so forth. The advantage of this, of course, was that you could pack a lot of information into a relatively small code. And if you're wondering, well, why would that have seemed so significant, keep in mind that most of the people that were working on this were studying small viruses, bacteriophages, or perhaps prokaryotes, that had very relatively small and compact genomes. And without knowing how many proteins were present, the idea was, well, probably nature tries to pack the information in as densely as it possibly can. And certainly, the information density in an overlapping code is much higher than the information density in a non-overlapping code. So maybe it's an overlapping code, or maybe it's a non-overlapping code. There's a pretty good reason to favor both. Of course, now that we know that the code is non-overlapping, less efficient way, but also much more robust to being able to just change one base, right? Remember, if we change one base in one of those codons, we could possibly change the amino acid sequence that it codes for, but it's not affecting the amino acids that are next to it. So nature kind of sacrifices that efficiency of packing things into an overlapping code with genetic robustness. It's less likely to disrupt the structure of the protein that you're coding for if you keep all the information contained with a single codon. So we now know that we have something called a reading frame. The reading frame is that sequence of 3 bases, which we call a codon, that are each calling out a single amino acid. It is typically at least typically around 50 of those bases, and very significantly, it always starts with an ATG. Okay, so ATG we'll see serves as the beginning of the sentence, if you will. Okay, starts with an ATG, which we'll see later codes for methionine. So that's the start. So if you're wondering, how do I know where the reading frame begins? You would find the ATG, the first one within your gene, and that is probably going to mark the starting position. We'll see that the start is a little bit more defined than that, but let's just consider now starting right there. Okay, so the next question is, great, so now we're going to guess 3. Turns out to be a good guess. We're going to guess non-overlapping. How do we start to actually map out what the code is? This is a really non-trivial problem, right? I mean, it's like literally discovering this language and nobody speaks it anymore because you obviously just can't ask. So this took a lot of very complex experiments, only some of which I can kind of illustrate for you, but at least I'd like to show you how one goes about beginning to assign the codes. And this is 2 different sets of experiments, 2 different groups working on it. Both use really clever strategies. Again, I can't show everything here, but at least I can show you how to start to assign the code. So the first thing that was recognized, and this was some pivotal discovery by Severo Ochoa, a very important biochemist, is that you can actually make template-free polymers of single bases, and as it turns out, mixtures of bases as well. But let's start with the polymers of the bases. So they generate, for example, poly-U. Okay, so you generate poly-U. What is poly-U? It's just 50 or 100 more U's in a row. It doesn't require a template. It's a polymerase that can do this. So he discovers this polymerase. And then he feeds that polymerase into the ribosome. And what does he make? He makes polyphenylalanine. So that must mean, if you're assuming 3, again, assumption, right? U must be phenylalanine. He does the same thing with poly-C. Feeds poly-C into the ribosome. What does he make? Polyproline. What does that mean? Proline must be CCC. At least just CCC, right? Does the same thing with A, makes polylysine. If you're asking why doesn't he do this with G, it's because if you remember all the way back in our discussion of DNA structure, poly G folds into these quadruplex structures, and it just doesn't, the ribosome won't take it. It basically folds up into something and it won't be accepted. Anyway, so these are the simple assignments, right? I mean, these ones are pretty straightforward. Then they start mixing in 90% U, 10% C, seeing all of the possible combinations of protein polymers that are being made versus those that aren't, and by changing the ratios of these different bases and seeing what possible peptides are coming out of those different combinations, they were able to statistically work backwards and figure out what the coding scheme would be. Does it sound really hard? Because it really was. Because remember, there were no machines back then for synthesizing these peptides they were making, right? These all had to be done chemically by hand. They had to make these, feed them to ribosomes, see what polypeptides came back, and then worked out statistically, well, if we put in 60% U and 30% C and 10% A, what are all the different peptides we get? And then as we vary these different bases, how does the base composition of the peptides change? And then working back statistically again to figure out the code. The other approach was a parallel approach that was also taken, was the idea that you could actually make short oligonucleotide sequences and attach them to ribosomes, right? So you could make short pieces of RNA, like mRNA light, attach them to ribosomes, and then make specifically radioactive amino acids that were attached to tRNAs. And so perhaps you made UUU and attached it to a ribosome. If you took radioactively labeled leucine, connected to its tRNA, and added it to that, it would not stick. It would just float away. So obviously, UUU can't be leucine. But if you took radioactive phenylalanine, attached it to its tRNA, right? Added it to a UUU, short nucleotide that was attached to the ribosome, it would stick. And so here's another piece of evidence that UUU must be the codon for Phe. So the combination of these two different sets of experiments, using these polymers and statistically working back what possible peptides could be coming out, depending on changing the ratios of the bases, and by using short oligonucleotides and determining which radioactive amino acids bound to tRNAs would stick to the ribosome, they were able to work out the codon assignments. I cannot still to this day believe that they did that, but this is what they came up with. And wow, they were right for all of them. I mean, this is not easy, right? Multiple years of work, many people now are doing it. Anyway, we now know that an open reading frame, as I've defined, so it's 50 or more of these codons, always starts with an ATG. And then it turns out that not only are there codons that are assigned to specific amino acids, but 3 of the codons are actually referred to as stop codons. These are where everything runs out and ends. So these don't have tRNAs that are associated with them, but rather they demarcate the end of the open reading frame. So this is what we call an open reading frame. Starts with the methionine, 50 or more codons. This is just kind of our definition. It's not a precise experimental determination. And if that's the case, it's probably going to code for a protein. So we call that an open reading frame. Anyway, what about this reading frame? What about those numbers? You have, by the way, the genetic code in the fact sheet, so that's not a problem. How unique is the code? Well, there are some amino acids that have a multitude of different codons assigned to them. You can see the most come from arginine, leucine, and serine. These turn out to be fairly highly abundant, not surprisingly, in proteins. If we look all the way at the opposite end, the least abundant of these would be methionine and tryptophan. 2 different good reasons for that. Tryptophan turns out to be evolutionarily considered to be a very big late addition to the protein game. It is in fact the least abundant amino acid in all of proteins, not surprisingly therefore that it has a single codon. Methionine keeps a single codon, basically because it represents the start. You don't want to have a couple different ways to start a sentence, right? In English we have a capital letter that designates it. Methionine designates the start of the open reading frame. So it also makes sense that nature would keep one single codon around for that. And then there are a smattering of them, okay? So these are the numbers that are different. Abundant amino acids tend to have quite a number. Rare amino acids have fewer. and then not surprisingly, methionine just has one. Again, it's going to represent the starting position. We always call this, by the way, the universal genetic code, but I just want to be clear that's mostly true, not entirely true. There are a handful of organisms that actually use distinct genetic codes. Here are some examples of them and how they change the coding. Keep in mind that this is, even though these are all kind of exotic creatures, our mitochondrial DNA also has a slightly different coding sequence. It's thought that this might evolve actually as a response to pathogens. If somehow an organism can survive a coding shift somewhere in evolution, it means that some sort of a virus that infects it won't be able to make functional proteins, right? Because it'll have the wrong codes. So even though we call it the universal genetic code, it should kind of be called the mostly universal genetic code because there are some exceptions to that. I've shown you a few here, but really you're also a very good exception because your mitochondrial genome uses an alternate code as well. Not only is that, but just quickly, given the fact that we now have a very good understanding of what the machinery that's responsible for attaching amino acids to the tRNA and the process by which those mRNAs get made, it's possible that artificially now, in the laboratory, we are not limited to only those 20 genetically coded amino acids ending up inside of proteins. Essentially what has been done is that scientists have evolved in the lab the enzymes that will attach pretty much any type of amino acid to a tRNA. And so if you were to make an mRNA with a code codon that's recognized by the tRNA, it will put in anything, right? So if you can get any sort of amino acid-like structure attached to your tRNA, it will add that. And so this is what we call genetic code expansion. The idea that we can hijack the code, and because the ribosome is not checking to see whether it's a correct amino acid, use this modified versions of what happens naturally to introduce amino acids that are not normally found in nature. And these are really exciting as experimental tools, but they may also be used therapeutically as well. There's some therapeutic proteins now where they are interested in using amino acids that are attached to radiolabeled molecules and so on and so forth. So pretty exciting, a bit of a side point, but still shows you that if you understand how this works, you can actually take advantage of it. Okay, but still we do have a small problem. And the problem is that 3 is a bit too much. We only have 20 amino acids, but there are 61 codons for it, right? So if you counted all the codons possible, took out the stop codons, there are 61 different possible codons, but only 20 amino acids. Remember, 2 wasn't enough, 4 was too many, 3 was also too many. Now, in principle, this doesn't have to really be a problem, right? Okay, so what this means is that even though you only have 20 amino acids, you only have 61 codons for them. And so at first guess, what this means is to cover those 20 amino acids, you need to make 61 different tRNAs. And by the way, that's not like impossible. I mean, nature could have just said, well, that's a lot of tRNA, but tRNA is already kind of the most abundant thing that you have, RNA-wise anyway, so that's fine. But it is kind of inefficient. Having to make 61 different types of tRNAs to cover only 20 amino acids is sort of problematic. And we'll growingly see what some of the problems might be, but right now you can just imagine that's like not really efficient. So ideally what we could have is a situation where we have, roughly speaking, the same number of amino acids for the same number of codons, or at least not so many, right? Because then we wouldn't have to make so many different tRNA molecules, and we wouldn't have so many different require so many different types of enzymes to attach those amino acids to the right tRNA molecule. So shrinking this down would be really advantageous. Advantageous because we wouldn't need as much different types of tRNA, and advantageous because we wouldn't need as many different types of enzymes to recognize the correct amino acid and attach it to the tRNA so the whole process could work. So shrinking that down would be something that nature would be sort of incentivized to do, and that's kind of what it does. Before we do that, there is another problem with this. It turns out to have the same solution, which is why I'm noting it now. What we will see is that a particular part of the tRNA, which is referred to the anticodon, is going to carry the information about what amino acid is attached, and it's going to recognize a part of the mRNA, which we've called the codon now, right? All fine and great. We needed to do so, of course, accurately. We wanted to get the right ones, right? We want to pair the right tRNAs with the right mRNAs, or we're just not going to get the correct protein. And so the best way to do that, we know, would be to have a very, very, very specific interaction. To make these these bases that are in the anticodon recognize the codon really, really precisely. That should be our goal. Except the problem with that goal is we kind of want to get around to making a protein. And remember, we already talked about this idea before in biochemistry, more than once, that the way to get a lot of specificity between two different molecules interacting with each other, that is to ensure that the right two molecules are interacting with each other, is to have lots of molecular contacts between them. But the problem with having a lot of molecular contacts between two molecules is that high specificity usually comes with high affinity. And high affinity means you're stuck. The higher the affinity between these two molecules, the longer it's going to take to eventually break them apart. And yet, that means that the more specific or more precise we get in trying to make a protein, the slower the process is going to go. So we also need to find a compromise there, right? In other words, we need to make sure that the interactions between our tRNA anticodon and our mRNA are specific enough to get the message right, but not so specific that it takes so long to do so that we never get a functional protein out of it. So these are sort of the two challenges we have. We have the challenges of having too many codons for just 20 amino acids, which means we need to make lots of more tRNAs and lots of more enzymes to attach the right amino acids to those tRNAs. We also have the problem of this is just not going to go very fast, and we need those proteins. We're not wanting to wait around for a long time. Anyway, the solution to those, as it turns out, comes from something we call wobble, and wobble depends at least in part on this deamination of this particular base here, adenosine. So we've seen deamination before happening. We've seen it happening in the context of DNA, of course, either unintentionally. We've also seen it happening in the course of making a variety of antibodies. Here, what it's actually being used is to make a functional base, and so an actual enzyme is going to do that. So we see here that if we deaminate the adenosine, we end up with inosine. Why do we care so much about inosine? Well, you will notice that inosine, not necessarily by perfect Watson-Crick base pairing, but can form base pairs with 3 different bases. So now we have a base that's not very specific. It's not very general. It can't base pair with everything. It's not very specific. And so inosine is going to play a big part in this phenomenon that we call wobble. Wobble is going to be the solution to both of our problems. Shrinking down the numbers of tRNAs by allowing single tRNAs to recognize multiple different codons that code for the same amino acid, and also by not being so specific and not making such great hydrogen bonds that we have to wait forever for them to break. So here again is the idea of wobble. Rather than have one codon recognizing one tRNA, we are going to have, in many cases, one tRNA being able to recognize a multitude of different codons as long as those codons are coding for the same amino acid. So if we look here at the codon-anticodon, the pairing between the tRNA and the mRNA, the first thing we notice is that it's anti-parallel. I'm making such a significant point about this because the number matters, right? The third base in the codon is the same as the first base in the anticodon. This anti-parallel arrangement is what gives rise to the numbering. Like when you mean first, Dr. Astrov, you mean the codon or the anticodon? So I'll try to be specific about that, but just to be clear, the third base in the codon is the base that in the codon we are calling the wobble base, but it corresponds to the first base in the anticodon. Okay, so that's an important point when we're considering the numbers. Now, if we look at the codon-anticodon pairing, looking at the anticodon of some of these tRNAs, right? We look at the anticodon of some of these tRNAs. Notice that if you have a C in that position, the only thing it can recognize on the codon is a G. Okay? The other two can be variable depending on what tRNA it is, but it's only a G. Similarly, in A, in the anticodon, the tRNA is only going to recognize one codon there, which has a U in its third position. That's it. So no additional broadening there. But if we go to the opposite end, we can see that if the wobble base in the anticodon is an inosine, it's capable of recognizing 3 different codons. One with an A, one with a U, and one with a C. That just jumped by 3 times, right? right? Instead of having 3 separate tRNAs to correspond to whatever is in X, whatever is in Y, A or U or C, we only needed a single tRNA for all of them. And so you can see that in some cases, there is a single tRNA will only recognize this particular codon. Sometimes it will recognize 2 codons of the same tRNA, differing only in the 3rd base of the codon. And some tRNAs, those with the inosine, can actually recognize 3 different bases that are in the wobble position, that 3rd position of the codon, further shrinking down the numbers. And so this is the idea, again, of wobble. Using in the 3rd base, the first 2 bases are conveying most of the information, I should say, about what amino acid you are, and the 3rd is like just kind of confirming it. It doesn't require the 3rd base to be accurate, right? It can make multiple types of recognition. It also isn't as good of an interaction. And so you can see here now, we've solved 2 problems. We've shrunk down the numbers of different tRNA molecules that are necessary to recognize a single codon, at least in many cases. But we've also weakened the interaction, not a lot, but just enough that those bonds can actually form, get the first 2 hydrogen bonds in place, get all of that, kind of test the 3rd one out, and if it's good, leave that amino acid behind, right? So the 3rd is conveying less information. It's there mostly for redundancy purposes. And so we have shrunk the number of tRNAs that are required, and we've shrunk the strength of the interaction between the tRNA and the codon that's required, meaning that this can happen efficiently in numbers and much more rapidly than if we had to make 3 perfect sets of hydrogen bonds between all of the bases in the anticodon and all of the bases in the codon. And so that's essentially the position of wobble, okay? Do you have to memorize the wobble table? I mean, no, I'm not asking you to do that, just so we're clear. A lot of people will go home and start, like, don't. I'm not asking you to memorize which ones are, but you should understand where the wobble base is, that it is inosine that gives you this high promiscuity in terms of what bases, and what 2 problems it solves and how to them. Okay, that's really what I would like to expect that you're able to do with this information. Anyway, the wobble doesn't change by very much, right? The 3rd base pair is very unlikely. So if there's a mutation in the 3rd base of the DNA that ends up inside of the mRNA and then gets translated, most of the time it does not change the amino acid sequence at all, so these are called silent mutations. But what about the other ones? What about at the opposite end? Is the code set up in some sort of intelligent way, or are those first 2 bases completely random? Was a very good question to ask. And it turns out that, look, it's not possible to guarantee that the first base isn't going to change anything, right? If you have a mutation in the first base of the codon, you're probably changing the amino acid sequence. But it turns out that the way that the code is set up is quite intelligent. That when you make first base mutations, most of the time, or at least many times, you change one amino acid to an amino acid that has similar chemical properties. So for example, if you go GUU to AUU, you get an isoleucine instead of a valine, which I know from our discussion of protein folding is probably going to be bad. But it's certainly not going to be as bad as going isoleucine to aspartic acid or aspartic acid to valine. And so this is what we call the mutational resistance of the code. The first base changes are probably going to change your amino acid, but in general they're going to change it to an amino acid that is as chemically similar as possible, so they may have a less severe impact on the folding or function of your protein. Okay, that being said, let's go see and take what we've learned now and see how we solve these problems energetically and information-wise of the ribosome. First thing to know, we're going to try as hard as we can to keep our 3-state model in play. It's worked really well for replication. It's worked really well for transcription. Not quite as well, but we're going to try to force it into that model anyway. We are going to have to add 2 additional steps, so that's why they're in parentheses. The last one is outside of the confines of this PowerPoint and also this examination. That's the subsequent folding, any sort of processing that needs to be done like post-translational modifications and localization. We're also going to have to get the tRNAs attached to the amino acids. Strictly speaking, just to be clear, that's outside of the ribosome. That is done far, far away. Just like last semester when we prepared activated and protected amino acids and put them in a separate bottle, that's the way we can think about the tRNA process. So strictly speaking, the 3-state model of initiation, elongation, and termination is kind of correct, but we do have to activate, and then stuff happens to it afterwards. Anyway, schematically speaking, this is just showing you the steps that are going to happen. I will be clear, in this PowerPoint, we're really mostly 99% going to focus on prokaryotes. When we talk about regulation, is really when prokaryotic and eukaryotic start to vary by the greatest extent. So I've kind of shifted all the differences with eukaryotes over into that particular PowerPoint. But there are a couple things. Where they appear, I will try to point them out if they're really significant for now. But the chemistry is the same. What's going on, the steps in the order of things are the same. But just to be clear, there are a couple things that we're going to have to kind of call out later on that are eukaryotic specific. Anyway, look, we've got an mRNA. We have some tRNAs. They're all loaded up. We have to bring everybody together. The mRNA has to find its correct position on the ribosome, which, although it consists of many, many proteins and ribonucleoproteins, normally is found in two separate parts. So after we get our first tRNA in there, that's going to carry our methionine and our mRNA. The large subunit is going to come in, seal this together. This is all kind of our initiation steps. Then we're going to start bringing in the correct tRNAs. They have to recognize their correct positions on the mRNA. As that's happening, the mRNA is moving in one direction. The growing ribosome is kind of, you can think about translating in the opposite direction there. And then, of course, the chemistry is happening, causing those two amino acids to be joined together. You just kind of keep repeating this process until you reach a stop codon. We'll see some proteins called termination factors will come in, cause the process to become disassembled, and then the ribosome can get used again, the tRNAs can get used again, the protein can go and fold up and get post-translationally modified. Anyway, this is just really the kind of the schematic of the whole thing. Just to show you, and maybe to validate the idea that it's okay to kind of talk about eukaryotic and prokaryotic at the same time. Obviously, because of regulation and because of more proteins being associated, the eukaryotic ribosome has more S's, right, which are kind of using it in circular size. But both of them are divided up into two subunits, a large subunit and a small subunit that consist of a multitude of ribonucleoproteins. If we look at the RNAs, again, if you just kind of look at the secondary structures, they're pretty well conserved. There's a little more complexity in the main RNA that's doing catalysis here, but there's a fairly high level of conservation between bacteria, all the eukaryotes. And again, just to be clear, this thing is massive, right? 18 nanometers wide, 2.7 megadaltons when the whole complex is assembled. That doesn't even include some of the proteins that are just coming and going that aren't remaining dissociated. But first, let's take a look again at these tRNAs, right? Because that's going to be critical to understand the tRNA process. We've talked a little bit about that before. This is obviously a heavily modified, many modified bases. The most common of which are the dihydrouridine and the pseudouridine that must be found in specific positions. Those are going to be important by recognition for other proteins. Notice that we are going to attach our amino acid all the way at the top 3' end. That's quite significant. That sequence that's there was not there, you'll remember, in the pre-tRNA. It actually was added by a separate enzyme. So the original sequence there that was truncated away, and those bases were actually put into place. And that's where we're going to go and attach our amino acid. All the way down at the other end is where we're going to find our anticodon. This is the one that's going to have the bases that are going to be recognized by the mRNA. So much like Crick suggested, these things are pretty far apart, right? On one end, we're going to have all the amino acid and the information there, and on the other hand, we're going to have the specific mRNA sequence being read. So whatever's attached up there really can get added. It doesn't have to be the correct amino acid, so some effort's going to have to be taken to make sure that in fact it is the correct amino acid. So now let's look at this. How do we get the process started? And remarkably, here's where we're going to use our ATP. So this is going to solve our problem. Now, before we look at the various steps, I want to be clear, because this is something that perhaps in the past I've not been clear about. I'm showing you some general chemistry, but the chemistry is catalyzed by a family of enzymes that are known as aminoacyl tRNA synthases, and there's one for every amino acid tRNA pair, okay? So this is not one enzyme that does this for all the tRNAs, right? Alanine has a few of these, depending on how many codons it has, I think 3, right? Cysteine has these. Methionine has just one of these. Okay, so the chemistry is general, working the same for all of them, but just to be clear, the amino acid we're grabbing is not like one particular machine puts them all on independently what it is. Each one has a protein associated with it. Anyway, so here we go. We're going to solve our little bit of ATP mystery now. Remember, we need to use coupling. The ribosome doesn't accept ATP as an energy source, and here's kind of the answer where we get our leaving group and our thermodynamic energy. Because the first thing that this is going to do is going to take our amino acid, the correct amino acid, here recognized by an R, take an ATP and adenylate it, and make the AMP, right? So adenylate it. Remember this chemistry? Remember ligase, how ligase used AMP as a good leaving group? Fundamentally speaking, this is the same chemistry, right? We're going to activate the amino acid by giving it a good leaving group. That's what we've done with every particular amino acid we are. Thermodynamically, what's driving this forward? Loss of the pyrophosphate. Thermodynamically, where's the energy of peptide synthesis coming from? Losing that pyrophosphate, destroying that pyrophosphate. Then all the energy's done. That is it. You have now inputted the energy, or if you want to think about this in terms of organic chemistry, added the good leaving group of making a peptide bond. The thing is, the ribosome doesn't accept that either. So even though we have made a good leaving group, we have not made a leaving group for a molecule that will be accepted by the ribosome. These adenylated amino acids are not, in other words, substrates of the enzyme that we need to act on them. So we need to transfer them to another leaving group, a leaving group that the ribosome will take. And that's really the amazing cleverness of the adapter hypothesis. Because the leaving group that's going to preserve all this energy, maintain the good leaving group, is tRNA. So you see, tRNA, yes, of course it is important, right, for recognizing the codon. I'm not saying that's not its role. But in terms of energetically, its role is to serve as a good leaving group to replace the adenyl group. And that's the second step of the reaction that the amino-acyl tRNA synthetases are going to carry out. They're going to remove this one good leaving group and replace it with another good leaving group, which is going to be the tRNA. The tRNA's other role in the peptide synthesis is going to serve as the leaving group for the actual chemistry that's going to take place. Now, there are two different classes of this. I have no idea why that is. I wish I could tell you. I don't know why, but one of them does the chemistry a little more complicated than the other, but they're both going to end up with the same result. So let's start by first looking here at the class 2 synthase, because that actually one is the most straightforward. The 3' hydroxyl is going to attack the carbonyl group. The AMP is the leaving group. All the energy is preserved. Here's your good leaving group. You have an amino-acylated tRNA. You're done. Okay, easy chemistry. In the case of the class 1 synthases, it's actually the 2' hydroxyl group that's going to do the attack. You might think, oh, so wait, I'm going to end up with something else. No, because those have a second active site that then is going to transfer the one on the 2, the amino acid. The nucleophile on the 3, the hydroxyl group, is going to attack the tRNA, which is attached to amino acid, effectively resulting in it ending up on the 3' anyway. Why nature has these two different classes, I cannot say. It does. Class 1, its first nucleophile is the 2' hydroxyl, and then the 3' hydroxyl acts as a second nucleophile, displacing the amino acid attached to the 2' hydroxyl. Whereas the class 2 just goes right for the 3' hydroxyl. There's not this intermediate step. The point is that all that leaving group potential energy that was present in the ATP, that was present in the adenylated amino acid, is now locked up here. We have an activated amino acid that has a fantastic leaving group, which means we can do nucleophilic chemistry on it. Now, before we go forward, there is a really additional critical step that's gone on here that is unique to the specific amino acid. The ribosome does not have any ability to check what amino acid is attached to the tRNA. All the chemistry is going to happen on one side. All of the reading the mRNA is going to happen far, far away from it. This thing is enormous, which means this is the last time to get things correct. If you attach the wrong amino acid, or something happens to that amino acid, an oxidation, a reduction, some modification for something that's floating around in the cell, that's going into your protein. And so there are really two critical features. One critical feature here, of course, is that there must be tremendous structural complementarity between the amino-acyl synthase, parts of the tRNA, and the amino acid. There has to be separate recognition that's very, very good, in fact exceptional, to recognize chemically similar amino acids, right? This is a real chemical challenge, you might think, not so much maybe with the polar amino acids, but valine and isoleucine don't chemically seem that different. There's nothing you can imagine an amino acid really grabbing onto to get it specific. So for cases like this, there are some proofreading domains that are present on the aminoacyl synthase, not on the ribosome, that are present on the enzyme that is attaching the amino acid to the tRNA. Proofreading really works by fit and size, largely, right? So where do you have proofreading? You have proofreading for particular amino acids that are not that chemically different. So for example, the valine is a smaller amino acid than isoleucine, right? But they are very chemically similar. And so if you had no proofreading, every so often you would have a valine ending up, an isoleucine ending up where there's a valine. So how does this proofreading process work? Essentially, it's based on fitting into the actual structure of the enzyme, or maybe hanging over a little bit. So valine fits nicely into the pocket where it's supposed to bind. It's smaller. Isoleucine hangs over a little bit, and where it's hanging over the enzyme just slightly corresponds to a nucleophilic site, where water can come in and cleave it off. So it gets loaded onto the tRNA and gets cleaved off because structurally it's a little bit too large. So look, not every single amino acid, the aminoacyl synthase, has a proofreading site. It's kind of hard to mistake cysteine for anything. Even though on paper cysteine kind of looks like serine, chemically the sulfur is very, very different than cysteine is. There's nothing that looks like there's nothing that looks like serine. So you don't really need to proofread for it. If a serine fits in and a hydroxyl group is fitting in the right place on the enzyme, that's good. There's not a mistake possible. But for where you have chemically similar amino acids, you have these hydrolytic sites where if it doesn't quite fit onto the enzyme as a complex with the tRNA, it just gets cleaved off by water. Okay, so we call this process the third genetic code, the correct recognition. The question is, where is it recognizing? It turns out that by doing studies of the tRNA, roughly 10 bases or so are contacted by the aminoacyl synthase, and those 10 bases are distinct enough to say, yes, this is the right tRNA for the amino acid that's fitting onto this side. So it's roughly about 10. Remarkably speaking, because only 10 are required, even though that is the correct tRNA for alanine, if you were to, in the lab, make that little mini tRNA only preserving those 10 positions, specifically that GU, the enzyme will still load it up with alanine. So it really doesn't take many, many contacts, as long as those contacts are highly specific, to ensure that the correct amino acid is actually there. Anyway, this is what we ended up with. This is where we are now. Class 1, class 2. Again, different enzymes for doing so, but this is the generic structure of the aminoacyl tRNA. And we are ready to make some protein. First thing we have to figure out, now that we have an mRNA loaded up, presumably, is where to start. Now, we said we're going to start at AUG, a methionine. The thing is, there could be a whole bunch of methionines in your protein, right? I mean, even though methionine is not the most complicated of the amino acids, abundant, I should say, you could have a couple. And how do you know which one's the right one? Do you start here? Do you start there? We might say the first one, but how does this enzyme know what is the first? The mechanisms here do differ a little bit between prokaryotes and eukaryotes, so this is one of the distinctions. In prokaryotes, the idea is that the ribosome is going to find a sequence, highly conserved sequence called the Shine-Delgarno sequence. So this is going to be a sequence that is appearing 5' towards the first start codon. And so the ribosome is going to find the Shine-Delgarno sequence and then start coding at the first AUG after that. The question is, well, okay, what does that mean to find the correct Shine-Delgarno sequence, right? I mean, you could write a highly conserved sequence out, but that doesn't mean it tells us anything mechanistically about what's happening. Well, what you see here is a piece of one of the ribosomal RNAs. That's the U-shaped one here. And you can see below it highlighted the Shine-Delgarno sequence of this particular message. And what the ribosome is actually going to do is slide this back and forth until you get good base pairing interactions with the Shine-Delgarno. So the mRNA comes in and it's sliding and wiggling and sliding and wiggling. When you have good base pairing interactions between these two, that's enough to stop it. That will position the AUG in the right place on the ribosome to accept the first tRNA. It will position it in a specific place. So you're going to find the Shine-Delgarno sequence by pairing up with one of the RNAs that are part of one of the ribonucleoproteins that make up the ribosome. That's how you're going to get started. The Shine-Delgarno sequence is, by the way, the conserved one on your fact sheet. So you don't have to memorize it if you were wondering. By a slightly different mechanism, but still similar principle, eukaryotes do not use a Shine-Delgarno sequence. They use something called the Kozak sequence. The principle is similar enough that we can lump them together. This is the Kozak sequence here. You can see that unlike the Shine-Delgarno sequence, for example, the ATG actually forms part of the codon recognition, the Kozak sequence. This is the sequence to find the right ATG. So start coding from here. Okay, this is the conserved sequence. Shine-Delgarno in prokaryotes and eukaryotes, it's actually called the Kozak sequence. Okay, so we have to get started. We know that a methionine is going to be first, but it turns out it's not going to be any methionine. Now, just to be clear, even though there is only one codon for methionine, in a complete contrivance of what I just told you about trying to shrink the tRNA numbers, there are actually two different tRNAs in prokaryotes for methionine. One of them links up with methionine. And for almost any methionine that's present in your protein is going to use that codon. But the first one, the one that's going to serve as what we call the initiator codon, is actually going to recognize this amino acid here called N-formal methionine, which is made by taking N-formal THF, so here's our THF group that we talked about, with a formal group that has been scavenged for some metabolic pathway we'll talk about later, attaches it here. That's going to be the first one. So the first one is actually going to be in prokaryotes, N-formal methionine. In eukaryotes, it's actually going to be just methionine. So this is a prokaryotic specific thing here, okay? This particular N-formal methionine is going to be attached by the correct synthase to a specific tRNA, and that specific tRNA is going to be the only tRNA that's possible for starting the process off. Okay, so it's not just methionine, it's N-formal methionine. Anyway, let's do some at least initiation. What do we need? We're going to make a polypeptide, right? So we're going to have, of course, a whole bunch of loaded up tRNAs. We're going to have our two ribosomal subunits, the small is shown here. There are three different binding sites in here, the E, the P, and the A site. All three of those sites are the right shape. It's kind of not really great in this figure to fit a tRNA in. So that's going to be the case. Any mRNA will do. We're going to need some initiation factors as well. These are specific proteins. GTP for some energy, magnesium to help us carry out some chemistry. Here's what I told you, more energy invested in getting the right amino acid than in making peptide bonds. None of that energy from that GTP is going into a peptide bond. That is all fidelity-based energy that's going to be burned here, okay? This is to get the conformational transitions right, the right tRNA to the right place. So you're going to see we're going to burn a lot of GTP, which is an NTP equivalent, none of which is going to be actually used to make peptide bonds. We've already completed our energy input into the peptide bond making process. We do need to have, of course, our tRNA with a met on it. We also need a protein called IF2, and yes, that is one that you actually have to know. It is a G protein, so it's bound to a GTP. It has a GTPase activity, which means you got to do this process, because once that GTP gets hydrolyzed to a GDP, it's over. So this is a time-limited process here. This recruiter, otherwise known as IF2, is going to take our tRNA with FMET, so our special tRNA, with our weird methionine, and bring it to the ribosome. What is it going to do with it? Well, the ribosome is first going to bound to two initiation factors, heading us set up the position. The mRNA is going to be loaded by base pairing with the 16 sRNA, the one that holds our Shine-Delgarno sequence. And now we have set up a binding site, and for the first and last time in this process, where our tRNA with the MET can now sit into that P site. This tRNA is special. Every other tRNA, except for this tRNA, must first enter the A site before it can move into the P site. But because of the unique structure and the role of IF2, it can sit directly into the P site. When it does so, the binding of the IF2 to the ribosome is going to speed up that hydrolysis, so now everything is going a little bit faster than it ordinarily would. It's all in the right place, everything's in position. That's going to cause a conformational change, causing us to lose our initiation factors. That allows the large ribosomal subunit to bind, and we are ready to start making protein with elongation. Elongation also has ingredients. The first of these, of course, is the previous step, right? We have a tRNA with the F met in the P site. We have the A and the E sites are both empty. Everything is set up there. We're obviously going to need all of our prepared aminoacyl tRNAs coated with the correct amino acids, so that's something we made separately. We're going to need some elongation factors, and yes, you do need to know what those different elongation factors do by name in the process. And then we're going to just keep kind of doing it over and over again. If you want to call EFG the translocase, you can. It goes by both of those names in textbooks, so that's what that asterisk is here for. The process, which we'll start now and then wrap up next time, and really termination is quick, so we'll be done with that rather quickly, is the following. We need this protein called 2, EF2, elongation factor 2, another G protein. When it is bound to GTP, it is going to take any amino acid tRNA, and try to dock it into the A site. And because of the way statistics work, most of the time it's going to fail. There is nothing, in other words, that's going to bring the right tRNA in. It is bound to a GTP, though. It's another G protein, so it's only got so much time to do so. So it'll bind, wiggle, 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 codon, codon, anticodon, anticodon. Nope, wrong one. I got to leave. Try another one. Nope, not that one either. So this is going to continue. You're seeing it look like there's one happening here. It's going to keep bringing them in. If it sits there long enough, that GTP is going to get hydrolyzed and assisted by the ribosome, right? And that's it. That's going into the protein now. There's no process of leaving. So this GTP is serving as a fidelity/timer. There's a finite amount of time nature's given it to test out the possible hydrogen bondings. Can I make the hydrogen bonds with the anticodon? Can I maybe make that? Nope, I gotta get out of here. Maybe can I make it? Can I make it? If it sits there long enough, it will hydrolyze, and that's actually sped up by the ribosome itself, speeding it up. It gets left there, and now you have an amino acid in the A site and an amino acid in the P site. Not surprisingly, what's going to happen, you bring a nucleophile next to an electrophile, a nucleophile next to an electrophile, it's going to put a leaving group on it. You're going to get a nucleophilic attack, and we just made our first peptide bond. So we have a few more slides on this, guys. This will be the last PowerPoint for this lecture, but please, of course, come to the next one at least, because we need to finish this up in about 10 minutes, and then we'll start looking at the regulation steps. You could do most of this problem set already, okay? So from here on forward, you just got to keep doing this a bunch of times until you get the determination of the process. All right? Great.